Welcome everyone. You are joining us for Advancing and Extending the Spectral Range for Imaging in Fluorescence Microscopy. This webinar is presented by Excelitas Technologies and hosted by us at Photonics Media. Today, you're going to hear from Dr. Gerhard Holst and Dr. Kavita Aswani. Dr. Holst is the Senior Imaging Product and Application Scientist at Excelitas PCO GmbH. And Dr. Aswani is the Senior Application Scientist for Biomedical Products at Excelitas Technologies X-Cite. In this session, they are going to discuss the advances in fluorescence illumination and detection in biological microscopy. You can ask questions or leave comments in the chat box at any time. If you have any technical issues during the event, please log out and back in to rejoin. You will be able to access recording of this online after the event, and you will receive a link to that recording in your email inbox. So with nothing further from me, I'd like to first welcome Dr. Gerhard Holst. Welcome.
Thank you, Joanna, for the nice and kind introduction. And welcome all to our webinar. Can you see my first slide?
Yes.
Wonderful. As the title already states, it's about advances and extending the spectral range for imaging in fluorescence microscopy. And my part, our responsibility, is to tell you something about on the detection side, while Kavita will tell you more about the illumination and excitation of fluorescence. The new spectral range that we are going to present is the extension towards the shortwave infrared radiation. So that's the longer wave end of the visible and near infrared light. This is the overview of my part of the presentation. So first of all, I would like to repeat the introduction of the shortwave infrared spectral range and explain a little bit about the circumstances and the possibilities of the detection of this type of radiation. I will shortly present a new camera that we have been developing, which is able to measure in the visible as well as in the shortwave infrared. And then I would like to point your interest to some interesting applications in microscopy and multispectral imaging. But let's start. Let's come to the topic itself, the shortwave infrared spectral range. What is that? Well, I found this nice table from the European Space Agency that compares the size of objects with the corresponding wavelengths of radiation and frequency. And as you might see there, from buildings down to humans, insects, grains of sands, there are human cells and protozoa which are in the size of the wavelength of visible and infrared radiation. There's a short blow-up that shows you the small range from visible 380 nanometers up to maybe 760 or 780 nanometers. And subsequently, there's the so-called near-infrared range, which has become more popular in the last years because on a deeper, let's say, penetration of radiation into tissue or cell layers, that is very helpful to get more information out of tissue samples. Connecting to that, let's use it for now, 900 nanometers. There is the shortwave infrared, the midwave infrared, and the longwave infrared. And this presentation will be focused on shortwave infrared radiation, which is assumed to range from 900 to 2500 nanometers. In general, this is a typical comparison of the features or differences between visible and shortwave infrared. So the upper row shows images that have been recorded and obtained in the visible range of the spectrum, typically with a silicon-based detector, while the lower row with the red background shows the corresponding images in the shortwave infrared. Starting at the left side, we have two apples, which look nice in the visible. And if you properly filter, for example, around 1450 nanometers, which is the absorption band for water, you can see that there are bruises of the apple skin, and the accumulation of the water appears to be darker in the image. So that's a possibility to check the quality of food before it's packaged. This is more, let's say, a cosmetic argument, while on the next image from the left, the second one, you see beans. In the visible part of the image, you cannot separate the beans from pieces of metal, stone, or plastic, while in the SWIR image, it's pretty obvious. For these type of controls, like packaging of beans or rice or something, if there is something in this material in there that is not wanted, it can sometimes be easily identified and spotted and differentiated in the shortwave infrared. Another application example is silicon. In the visible, a silicon wafer is pretty intransparent. It's black. You cannot see through. So therefore, you can also not check whether there are some damages or blemishes within the material, while in the shortwave infrared, the silicon becomes pretty transparent, and there you have a possibility to look into it and check. Another potential application or difference in shortwave infrared is shown on the fourth image, which shows some soldering irons. You cannot measure precisely absolute temperature, but you can see temperature differences. And here you see a switched on soldering iron compared to another soldering iron at 100 degrees lower temperature. The advantage of the shortwave infrared to do this is you can see even through glass windows, which is not possible with an infrared camera. A final application is as well some of the dyes that are used to dye or stain plastic materials. They become very transparent in the shortwave infrared, and therefore it's a possibility to check whether boxes are filled and if, how much. So these are classical advantages or differences if you compare visible images with images that have been obtained in shortwave infrared. Here is a more recent example that we have achieved on one of the workshops that we've given on the Trends in Microscopy conference in Germany this year in March. And you see there a smartphone image with two objects on the left-hand side, a water bottle and a typical Thorlabs box that has a blue tape for indicating what is in there. This is the smartphone image in the visible range. And on the right hand, you see the corresponding shortwave infrared image that has been filtered with a long pass filter at 1000 nanometers. And you might appreciate that in the shortwave infrared image, the water turns to black due to the water absorption at 1450 nanometers. And funny enough, the blue tape disappeared. You don't see it any longer. So you can look through. But how can this be done? Well, if we go to the pure image sensors, then usually you need a material that can generate charge carriers with the energy that is given by shortwave infrared radiation. Typical silicon detectors, which are used for cameras in smartphones, in scientific cameras for fluorescence measurements, for silicon, the energy is not enough. Therefore, in all cases, a SWIR-sensitive image sensor is made of a sandwich structure. You see on the left-hand side a larger indium-gallium arsenide layer, which is able to have a band gap that is small enough such that the radiation energy of the shortwave infrared radiation is large enough to generate the charge carriers, while the charge carriers subsequently are then guided through these vertical bumps to be processed by a silicon readout circuit. On the right-hand side, you see some enumeration of manufacturers of these classical InGaAs image sensors. And due to this sandwich structure. The problem is you will have to position in the manufacturing process the InGaAs layers on top of these readout circuit with a high enough precision. To achieve that, in classical sensors, the bumps have to be large, and the pixels, therefore, the light sensitive elements of the image sensors also have to be quite large. Another possibility for SWIR detection as well are quantum dots. So this is shown here on a graphic that I've taken from the SWIR Vision Systems website because it's quite nice. Again, a sandwich structure. In this case, the quantum dots absorb the shortwave infrared radiation and generate the charges, and they are vertically connected to, again, a silicon-based readout circuit. The advantage of the quantum dot is in the manufacturing process, because you can deposit these different layers with a more or less simple spin coating process on a wafer, which makes the manufacturing cheaper, but also the quantum dots have some kind of disadvantage in terms of sensitivity. Now, what has generated with us the interest as well in InGaAs sensors is there is a new player in these manufacturer family, and this is a company named Sony, which has an enormous impact on the image sensor market because they are supplying between 45 to 47% of the total image sensors worldwide. Due to their experiences in manufacturing as well stacked sensors in the silicon area, they know how to use smaller of these bumps because they have the manufacturing experience. This allows to make smaller InGaAs sensors, and this in turn makes the InGaAs sensors a little bit, or not only a little bit, but significantly cheaper, because the main cost driving factor of a digital sensor is the area that it uses from a wafer. Therefore, now Sony has created new InGaAs sensors at a smaller pixel pitch. And again, due to that way of manufacturing, it allowed Sony to use thinner InGaAs layers, which in turn makes the image sensor as well sensitive in the visible range. So you get both worlds. Here you see some typical quantum efficiency curves. One example is the Sony, which is in this case a relative quantum efficiency. Later on, I'll show you some QE curves that we've measured with these type of sensors. And you see there's a sensitivity from 400 to 1700 nanometers, which is a broad range. In the middle, from Xenix, you see a typical QE curve for InGaAs sensors, which ranges from 900 to 1700. And on the right-hand side, there are two examples of colloidal quantum dot SWIR sensors, which show a broader quantum sensitivity, but as well a much lower absolute value in the range between 10 to 15%. And just to remind you, the new Sony sensors have a resolution of 1280 by 1024 pixel at a 5 micron pitch, while the classical sensors in InGaAs had 20 microns, 25 microns, 15 microns, and the quantum dot sensors are also in the range of 15 microns and 20 microns, quite large. A general large interest in this type of image detectors comes from automotive industry because they would like to use SWIR image sensors to see through fog and rain. Therefore, there's a lot of research ongoing, and here are some recent results that I've received from ST Microelectronics on a workshop in London about image sensors. And you see some of the sensitivities of the quantum dot sensors have been improved, but still they are quite low compared to the classical InGaAs sensors. We've used the new Sony sensors to create a camera out of it, which is called PCO Pixelfly 1.3 SWIR, with the following features. I mean, this is the Sony sensor. It has a broad sensitivity. Due to the lower dark current, it allows for somehow longer exposure times. And the camera is powered via the USB connection, and it supports the GeniCam software and API. Here you see an example from the laser show this summer in Munich, where we've demonstrated the features of the camera. I would like to continue with some of the results that we've measured so far. So here you see absolute quantum efficiency values of the Sony sensor that we were able to detect. And it's a very impressive sensitivity over the whole range. But, and this is something that I have to emphasize, if you use, for example, in microscopy application, scientific CMOS cameras and sensors, then the readout noise features of an InGaAs sensor are a lot different. Because if in the CMOS world you are comparing 1.3 to 1.8 electrons or 0.8 or 0.5 electrons readout noise, in the InGaAs world we are talking about 180 to 200 electrons. That's a lot more, which means for a proper image you need somehow a lot more light. As we are supporting the quality standard defined by the EMVA, which is called the EMVA 1288 standard, we are doing all QC measurements, even for the SWIR camera, compliant to that. And here you see some two typical examples, what is measured, the photon transfer curve, that as well can be used to derive the conversion factor. I mean, how many electrons are used to create the number in your digital image. And on the right-hand side, you see the linearity curve, which looks pretty good. So these cameras can be used for measurements. Which might be as well new to you are the type of applications that can be performed with these new spectral range. If I come now first to in vivo and intra-vital microscopy, for example, you see here an image of a mouse skull back illuminated in the near infrared. So you have an impression, yes, you can get light through that. And you see the emission of the ICG, I think they've used to for the blood system. And if you compare this to the image in the shortwave infrared, you might appreciate that you see much more of the blood vessels and a much better contrast in the image. And this is something that, for example, the working group of Professor Bruns, from whom I received these results, is working a lot with to improve that, because it can be used due to the lower scattering and absorption to look into the animals without cutting or opening them. Here you see some results they obtained at the different wavelength ranges, ranging from 1000 nanometers, 1200 nanometers, 1450 and 1600. If you look through to the graphs below the photographs, then you see that the signal also is decreasing. And I mean, it's not a surprise, because at 1450, there's the highest absorption of water, and there is a lot of water present in the tissue. On the other hand, what is also increasing is the contrast, because all photons that are generated and have to overcome a larger path due to the fact that they are scattered have a higher probability that they are absorbed. Therefore, mostly the directly leaving or emitting light from the tissue is detected by the camera. So it gives a better contrast, but you have to cope with a lower signal. On the next slide, you see the same images for a whole mouse. And again, you might appreciate that at 1500, you get the best possible insight into the mouse. This has been further investigated by Professor Bruns and his collaborators. And they have figured out that there are quite a few fluorescent indicators that can be used, which are excited at different wavelengths, but they all contribute in the same window some type or some amount of emission. If this is the case, you don't need different filters for doing multispectral investigations, but you use different excitation wavelength, and I guess Kavita will talk about that later on as well, but use the same detection window. So you could do with this kind of a spectral multiplexing. And on the next slide, you see the result in a living mouse. I hope you can see the movement and the 3 different indicators, and you can see inside of the mouse without cutting or opening it. Further investigations have shown this can be done as well without fixing the mouse. If I start the movie right now, you see that this mouse is not fixed, it's just observed, and it can move. And even better than that, well, this is, sorry, this is the one paper dealing with different fluorophores, because currently there's a lot of research and investigations are ongoing, because all the nice features of SWIR don't help you if you don't have proper stains. On the other hand, there are already some stains that exist, like ICG or like IRDI 800, and they can be modified to couple to different or to stain different molecules and for different investigations. And here you see more recent results. And I start now the movie, and this movie honestly impressed me a lot when I've seen it first, because these are free moving mice, and the organs that have been stained are very good visible. And therefore this will open, in my opinion, a lot of possibilities for future research in cancer. So these were the applications of intra-vital and in vivo microscopy. And for example, these were some results that we obtained with our camera with a frozen dead mouse. But again, the 4 images combined or merged into one color image show maybe not so impressive, but a very similar result. Another possibility to use this type of multispectral approach is maybe not fluorescence, but absorption and reflection. We've used such a more industrial microscope made by our colleagues from Key Optic, and a ring light that we created with different wavelengths. And using the camera, we can analyze the multispectral data with different methods, typical principal component analysis or singular value decomposition, as well as some supervised learning feed methods like partial least squares, discriminant analysis, or random forest classifier. We've put this into a software that can be used. And as an example, I would like to show at the end of my part of the presentation this principal component analysis. And we've recorded at different wavelengths pictures of 3 different leaves and tried to analyze them. And here you see the different principal components based on the assumption that we are assuming 6 components being present. And now if we have done this analysis, we can create so-called score cross plots that show the similarity and dissimilarity of the different components with each other. And each of these pixel clouds represent a certain pixel in the original image. And this means that if we mark or have a look where these pixels are in the original image, we get some information back from these score cross plots. Therefore, we can draw rectangular or ellipsoids into these score cross plots and ask ourselves, well, to which pixel in the original image correspond the pixels within these frames? And you see in this result, with 3 different of these frames, we are able to identify the different leaves. Now, this result should be independent of the position of the leaves in the image. Now, if we take some more of these samples, then we only need to look into the pixels that end up in these frames. So that's one possibility of analyzing these multispectral data. And we're happy to provide this free software with a camera if it's going to be tested. So if you have interesting applications that are relying or relating to multispectral investigations, we are happy to give it to you for testing. This is pretty much the end of my part of the presentation, and now I would like to hand over to Kavita to tell you which type of illumination can be used for that purpose.
Sorry, I was muted. Thank you, Gerhard. Can you see my screen?
Yes, looks good.
Okay, thank you. So, so Gerhard covered the detection side in extending the spectral range, and I would like to briefly discuss the illumination side and the options that are available for illumination, both in the kind of near infrared, but also to the left side of the curve, which is in your near UV. So I'm going to turn off my camera so we can focus on the actual presentation. As far as illumination is concerned, obviously that's important. You do need a good, stable, uniform illuminator in order to be able to get data that is quantifiable and that is accurate. If you have uneven illumination, obviously you'll have differences in intensity in your slide or in your tissue or even within a specific cell. So I just want to briefly introduce the X-Cite illuminators. So what is X-Cite? We were the first alternative to this gray box that you see on the top right here. And with this gray box that was attached to the back of the microscope, you would see that this is the actual bulb that was used for this technology, which only lasted a few hundred hours. Of course, since then, things have moved on to LED, but X-Cite was the first to replace this gray box with a bulb that is pre-aligned that you just snap into the actual unit and you're good to go for at least 2,000 hours, sometimes 3,000, sometimes 4,000. Since then, obviously, we have moved on to LED technology. And just to cover the range of what's available in LED technology, there's essentially white light systems, and then there are switchable systems. And by white light, that means a broadband source where you would have to do the filtering in the actual microscope. In the switchable options, you would have filters built into the unit, and you would thereby be able to much quicker switch between the different wavelengths for slide scanning, for multiplex imaging, for high throughput screening. So the one I'd like to focus on today is the X-Cite Novum, to kind of pair with the whole extending the spectral range of illumination, as well as detection, as Gerhard covered. So the X-Cite Novum, which was launched maybe a couple of years ago, and this is the key to the Novum, is that we can go from 340 nanometers, which was used for fluoro imaging, where traditionally you've had to rely on xenon lamps, which are very, very big instruments, very, very expensive instruments. And it goes all the way to 800 nanometers, which would help excite ICG, for example, which is a fluorophore that Gerhard covered in his, which is closer to the near infrared. So the Novum is our newest X-Cite product. It has 9 high power channels. You can switch between the wavelengths for fluorescence imaging. It does also have what we call our laser LED hybrid drive. And what that is it allows you to get a lot higher power in the green gap, which is where LEDs have traditionally suffered compared to lamp. So we have our proprietary technology that enables high power between the 500 to 600, previously known as the green gap. The unit also has cleanup filters so that you can narrow the wavelengths of interest before the light leaves the box. It's very easy to control. You have your typical USB control using third-party software drivers, and you can also do TTL triggering as well as analog triggering for all the 9 colors. And there are 4 configurations available, which I will briefly cover just so you know the options that are available. So these are the 4 different options that are available with the X-Cite Novum. It would cover your DAPI, CFP, FITC, and then you have your YFP, M-Cherry, Sci5, and Sci7. So all the flavors will include Sci7, which is fairly new as far as being available in multi-wavelength systems. We then have the Fura option, which here is in yellow. And for the Fura ratiometric calcium imaging, you require a 340 and you require a 380 nanometer wavelength. You then end up taking an image at 340, another image at 380, and then you take a ratio of the 2 to figure out the change once calcium was introduced into the sample. Finally, the IR version, which is here in red, that extends all the way to 800. And as mentioned, IR is important because ICG is a fluorophore used in surgery, which is actually FDA approved. So a lot of people in labs are extending their research into the IR region in order to be able to do better understand how it's being used in surgical applications. So the gold standard in fluorescence imaging has been either xenon or metal halide or mercury. So here in the yellow, you see our own X-Cite mercury lamps, metal halide lamps, which we still offer, but most people want to move to LED. And in order to get the LEDs to have the same power as the metal halide, we've had to get creative and patent technology that gives you the bar in the green. And you can see that at all the popular fluorophores, the bar in the green has exceeded our own metal halide lamps. And then the bar in the blue is the Novum, which I have been discussing, and you can see that it far exceeds that of the metal halide. So where would you use the Novum? So depending on the application, there's different flavors of the Novum that can be chosen. You have your fluorescence in situ hybridization, multiplex imaging, Fura calcium ratiometric imaging would utilize the Novum Fura. Your far red excitation and the Sci7, we have 3 different flavors that can be used for that. And for the near infrared excitation at 800, you would use the Novum IR. So very briefly, and I have covered this already, the laser LED hybrid drive boosts the power between the 500 to 600 to fill in the traditional green gap. So just some sample images of Fura 2 calcium ratiometric imaging. You would take you would take an image at 340, you take an image at 380 of the control. Then you would add something that would cause a calcium influx. And then you would take another image at 340 and another image at 380. And that's your ratiometric imaging that's used in Fura. And then you can see the difference that was caused between baseline before the calcium and then after the calcium. So basically, again, you don't have to use big bulky xenon for this, you now have an LED option along with a whole bunch of other LEDs within the same unit. FISH, which is fluorescence in situ hybridization, as you may know, is targeting very small sequences of DNA or RNA. And because you're targeting such tiny molecules or oligos, you do have a very weak signal for FISH. So in order to be able to image these weak signals, you would require a high intensity light source. And this is just an example of how the Novum, with its very high optical power, can enable you to perform FISH successfully. And then, of course, with the IR, and Gerhard has shown you some amazing images of this, but the IR allows you to image deeper into the tissue, in vitro or in vivo. And because it's infrared light, it penetrates deeper into the tissue with higher signal and lower background noise. And as I mentioned, it is gaining popularity in IR imaging because of the fact that surgery is moving there because ICG is FDA approved. Finally, the X-Cite Novum was tested for a spinning disk application. And you can see the wide field video here, which is fairly decent. But then if you attach a Novum to a spinning disk confocal, you can see how much more crisp the image is. Sorry. You can see how much crisper the image is when you use spinning disk confocal. So if you'd like to get more information about the Novum and how it works, we have a whole bunch of videos and resources available online. And next slide. And that's it. Any questions relating to the imaging, illumination, or detection?
Yes, thank you so much, Kavita and Gerhard. We do have time for a few questions now before we close. Everyone, if you have not already, please go ahead and type those questions and comments into that webinar chat there. All right, so let's see. The first question I have is, do I need special objectives to apply the camera in the SWIR spectral range?
I guess that's on my side for the cameras. And yes and no. Certainly you need specific objectives in microscopes or C-mount objectives that are properly corrected for that spectral range. And there are only few objectives that are corrected for both ranges, from visible to SWIR. And only these don't have to be refocused. Otherwise, you have to live with the fact that when you change the illumination from, let's say, SWIR to NIR to visible, then you will have to refocus to get a proper focal plane on your camera.
Great, thank you. All right, let's see. This next question is, how can I use the camera with my own software?
Well, that's relatively easy. We offer a free software development kit, as well as support for some of the classical, let's say, third-party open source software packages. So there will be a support for Micromanager, and maybe we'll get some support by the software packages offered by the microscope manufacturers. We are working on that. And as well, there is the software that we offer for free for the multispectral analysis, because this allows to control the camera as well. And we also have a free camware that can handle all adjustments and all settings and operational mode of the camera. And you can store the images and then use whatever image processing package you would like to use.
Great, thank you so much. Let's see, this next question is, how can I trigger the light source if I want to do multispectral imaging with different illumination colors?
So as mentioned, you can, there's obviously the USB control, there's manual control, but there's also a TTL option. so that you can send voltage to each of the 9 colors, time it the way that it's required, and you can then trigger it that way. Or obviously in any of your software packages, that's not trigger per se, but you can have it programmed so that it turns on and off by USB according to the timing that you want. And then obviously the camera will do its job as well through the same software.
Great, thank you so much. And I think that's all the time we have for questions now, but everyone, if you do think of another question, you can always send us an email. You have Kavita and Gerhard's emails there, or you can reach out to webinar@photonics.com. That's webinar@photonics.com. And just one final note, the whole session was recorded. It will be available on the Photonics Media website shortly, and you will receive a link to that recording by email. So I'd like to thank all of you for participating today and thank Drs. Gerhard Holst and Kavita Aswani one last time. Thank you both. So this webinar was
Thank
presented
you
by
for attending.
Excelitas and hosted by Photonics Media. Thank you, everybody.